Welcome to Backbenchers. I hope everything is going well with you all and I hope you all are fine. In today's video, we are going to be discussing about the fourth part of the chapter Absorption by Roots under the playlist of Plant Physiology. We have already discussed about the first three portions. So if you haven't seen the videos yet, then please go and check them out. I will definitely provide the link of the playlist in the description box so that you can see the videos easily. Let us quickly have a look into the division of the chapter. First was the introductory part, which we have already finished. Second was the roots and its advantages, which is also done. And third was the osmosis and its types. In today's session, we will be discussing about terms of osmosis and its significance and tonicity. And then these are the rest of the portions which will be done accordingly. And I have also mentioned before as well that after finishing all of these portions, I have kept a separate doubt class for you all. So whatever doubts you're having, please do not hesitate to write it in the comment section, because I will be answering all of your doubts at the end of each chapter. I'll shortlist the doubts which are most frequently asked and most common ones, and then I'll answer them in that particular doubt video. Apart from all of these portions, I have also organized a PDF for you all. That PDF is going to hold a lot of important questions, a lot of MCQs and a lot of short questions as well. I will provide the PDF at the end of the chapter, that is after completing all of the segments of the chapter. I'll upload the PDF in the description box. If any one of you wants me to check your answers, then please mail me your answers. I will be providing the mail ID as well in the description box. So please stay tuned for the PDF and at least go through the question once of the PDF, because the PDF is going to hold a lot of important questions. So let's not waste our time any further and let's just start with today's portion, that is terms of osmosis and its significance and tonicity. It's a very easy portion that we have today, and it's a very fun portion as well. You all will be able to understand it very easily. First, let us look into the terms of osmosis. Okay, before going into today's portion, we have some questions from the previous session. Let us have a look into them. If you want, you can answer them in the comment box. I'll check them out for you. The questions are very easy. Go through the question once. We have already discussed this in our previous session. The first question is, which part of the root cell performs osmosis? Very easy question. While teaching osmosis in the previous session, I have said this thing to you all very clearly. So if you remember, you can answer it. And if you have a problem to answer this question, then you can go and check the last video once again to clear your doubts regarding the last question. Then the next question is, what is the basic difference between osmosis and diffusion? Again, this question is also being answered by me in the previous video. It's a very simple question I've given. So please make sure that you remember the answer of this question without any problem. And if you have the problem, I've already said that please go and check the last video again. So these two questions are from the previous session. Now let us move on with the today's portion. Okay, so we will be discussing about the terms of osmosis now. The first term that we are going to study today is osmotic pressure. Now we are going to be understanding what is osmotic pressure with the help of a very simple experiment. This experiment is already known to you all. If you remember that the previous session, when we were doing the experiments regarding osmosis, in the first experiment, we used thistle funnel to demonstrate osmosis. And this is the same experiment that we are going to be taking the help of today. So this is the same experiment of the thistle funnel, and we are going to use this experiment to understand the concept of osmotic pressure. You can see that the apparatus is more or less similar. We are going to be just adding a single different thing in this apparatus. But first, let us discuss the apparatus and the things taken here. This is a beaker. This thing is a beaker, and the beaker is containing water. So water is present in the beaker. Let me just write it beside water so that you can understand. Next is this sugar solution. This is basically a round bottom flask. I'll just write it in the side of the round bottom flask. Round bottom flask, yes. And then this is the thistle funnel of the round bottom flask. Then this is the semi-permeable membrane. Semi-permeable membrane means there we are using cellophane paper. Cellophane paper, all of you know cellophane paper acts as a semi-permeable membrane. So we are just using cellophane paper here. We have taken sugar solution here. This is 20% sucrose solution. Make sure you remember that as well. This is the initial level of solution where we will just mark it in this particular way. Now, the only new thing that we are going to be adding in this apparatus is a piston. Let us draw a piston. A piston looks like somewhat like this. I cannot draw it perfectly though, but it will be easier for you to understand if I draw it like this. So this is the end of the bottom of the piston, which is inserted and just placed right at the level of this solution, at the top of the level of the solution. And then the piston has a neck like this, which comes out of the thistle funnel, and then at its top, wait just a second, let me just do this portion again. Yes, okay. And then at the top of its the piston holds a weight. A weight is present here. To show the weight, I'll be using a different color so that it becomes easier for you to see it. Here, a weight is placed at the top. This thing is the weight. We will just quickly mark all these things and then we will get into the explanation of the whole experiment that is taking place here. So firstly, we will mark the weight here. This thing is the weight. And this thing is the piston. You don't have to be scared about the term piston because it is a new term. For understanding this experiment, all you need to know about piston is a piston is something, or you can say a piston is a device, that is movable. This movable nature of piston is going to help us understand the concept of osmotic pressure. So this experiment we are going to be performing just like the previous experiment of osmosis, and then we are going to be letting the apparatus sit for some time. Now, if we recall the previous experiment, when the apparatus was let to sit for some time, we observed that there was an increase in the level of solution inside the thistle funnel, which proved the existence of osmosis, right? In the previous experiment, if you all remember, the change in the level of the solution inside the thistle funnel was helping us believe or was helping us understand that osmosis is taking place. But in this experiment, in this scenario, with the entry or with the use of piston, there is no such thing happening. That is, osmosis is not taking place. So if we observe this experiment, this apparatus after some time, then what we are going to notice is the level of solution inside the thistle funnel and the level of water inside the beaker, both are remaining as they were. So, which means, suppose let us consider that this is 50 ml of water we have taken in the beaker. This is 50 ml. And inside the thistle funnel, we have taken 20 ml of solution. Just as an example, just for an instance. After some time, what we are going to see that the same configurations are still maintained. That is, there is no change, there is no fall, or there is no rise in any of the liquid in any of the places. That means osmosis is not taking place. Why osmosis is not taking place is the matter of concern. Osmosis could not take place because of this weight that you all can see right here placed at the top. This weight. Why? If osmosis would have taken place, then what would have happened? Water molecules from the beaker would have gone inside the round bottom flask, crossing the semi-permeable membrane, and then would have come here inside the thistle funnel, and then the level of water would have increased. Level of solution would have increased inside the funnel. But here, since the piston is present and the weight is applying a force on the piston, water molecules are not being able to move the piston upwards and increase the level of solution in the thistle funnel. That is, water molecules have to put a force to increase the level of solution inside the thistle funnel. So what is happening here? Water molecules are entering. Water molecules are exerting a force inside the liquid column of the thistle funnel. And an equal and opposite amount of force is being exerted by the piston over the water column as well. So this is more of like a physics concept. Reaction force is taking place. If we put a force on something, that thing puts a force back on us. Same thing is happening here. The piston is putting force over the liquid column inside the thistle funnel, and so as the water molecules doing the same thing, because the water molecules are trying to go inside the solution and to increase the level of water, but the weight is not allowing the piston to allow the further entry of water in there, thus preventing the osmosis to take place. This thing tells us about osmotic pressure. So what is happening? What we will notice after some time, that at a point the force exerted by the piston due to the overlying weight becomes equal to the force exerted by the water molecules to go inside the thistle funnel. Just as I said you all that this is a concept of physics, sort of like a reaction force. The force exerted by the piston over the liquid column is equalizing with the force exerted by the water molecules to go inside the thistle funnel. This force is known as the osmotic pressure. So what is this force doing? This force is not only just preventing the entry of water molecules, this force is also preventing the process of osmosis to take place. So this force, let me just write it here for you all. The force is preventing the entry of water molecules as well as preventing the process of osmosis to take place. Osmosis to take place. This is what is happening here. This force, which is, we are just going to be considering the force which is preventing the process of osmosis to take place, because osmosis is more important here. So the force which is preventing the process of osmosis to take place is the osmotic pressure. So it is the pressure. So we can say that osmotic pressure is the pressure which is required to prevent the process of osmosis. To prevent the process of osmosis to take place. That is all about osmotic pressure. Very simple to understand. This experiment actually helped us a lot to understand osmotic pressure in a better way. Now, what is the second term that we have is osmotic potential. What is osmotic potential? It's a very simple thing to understand. Once you have understood osmotic pressure well, you can very easily understand osmotic potential. Pressure develops in a closed system like a cell. Yes, suppose something is closed in a box like this, and it has no passage or no holes for the passage, no place where air can pass through. In this particular system, pressure is going to develop. Pressure is going to develop. So pressure is always developing in a system which is closed from all the four sides. Hence, in such a system only, osmotic pressure term is going to be used. For a system like the thistle funnel, which is open and not closed from the four sides, in that particular type of system, we are going to be using the term osmotic potential in place of osmotic pressure. Both mean the same. The only difference between the both is osmotic pressure is used in a closed system. Used in a closed system. And osmotic potential is used in an open system, like a thistle funnel. An example of closed system is also given here, which is cell. Example of open system is thistle funnel. Thistle funnel is also written here. Thistle funnel is also written here. So now you've understood what is both the terms of osmosis, osmotic pressure and osmotic potential. Let us just simply write these definitions once again to help them understand in a better way. First is osmotic pressure, the pressure required to prevent the passage of pure solvent into the solution when they are separated by a semi-permeable membrane. There was supposed to be a Y here. Okay. When they are separated by a semi-permeable membrane. Acha. You can define osmotic pressure in another definition. Let me just write that definition for you all. You can just simply write the definition of osmosis to define osmotic pressure. So what we are going to write is the pressure needed. In our own language, we are going to write this definition. So this definition is going to help you understand better the concept. The pressure needed to prevent the movement of only solvent molecules from the region of higher concentration to the region of lower concentration across a semi-permeable membrane is called osmotic pressure. It's a more simpler definition. You don't have to remember any other definition. You have to only remember the definition of osmosis properly. Then only you can make your own definition of osmotic pressure. And osmotic potential is also a similar thing like osmotic pressure, but we have defined here osmotic potential in a different way. We have written that the minimum amount of pressure or force required to nullify the process of osmosis is called osmotic potential. Now, what is the meaning of the term nullify? The term nullify means to prevent or to stop. This term means to stop. That is to completely, not just stop, to completely stop the process of osmosis. To completely stop. That means nullify. That was all about the terms regarding osmosis. So we have already discussed about the terms regarding osmosis. We knew that what is osmotic potential and what is osmotic pressure. The next thing that we are going to discuss is significance of osmosis in plants and animals. The process of osmosis has significance in both plants and animals, not only in plants but also in animals. What are those significance? We are going to be looking at the significance clearly. First, let us look in the significance of plants. The first point of significance is required during absorption of water from soil. We have discussed this plenty of times. We have known osmosis is the process in absorption by roots through which plants absorb substances from the ground, or roots absorb substances from the ground. So absorption by roots need the process of osmosis. So osmosis is required by the plants for the absorption of water and minerals. Not only water, and minerals as well. And minerals by the roots. Second important point in plants is required during opening and closing of stomata. Now this point involves the phenomenon of turgidity. We are going to be discussing about, okay, I hope the ink is not too bright. Let me just change the ink. It might be too bright. The color is contrasting, hence it is very bright as far as I think. Let me just change the color of the ink quickly. Okay, I think this is going to go well with the color here. Yes, okay. So required during the absorption by roots, absorption of water and minerals, and then it is also required during the opening and closing of stomata. Now this point involves the phenomenon of turgidity. We have not yet discussed about turgidity. We will discuss turgidity in our next session. Till then, you all just keep this point in mind that osmosis helps the plant in closing and opening of stomata. Let us look into the significance of osmosis in animals now. In animals, firstly, we have amoeba here. Remember one thing, amoeba is a scientific name. That's why I've written it in an italic form. But when you all will be writing a scientific name in your copy, then make sure that you underline that particular word, because it is very, very important. That's why I've written amoeba in this particular manner. Let me just underline that again, so that you don't forget that this is a scientific name and it must be underlined while writing your answers like this. So amoeba is a freshwater animal, and hence it maintains its osmotic balance by letting water enter into its body through osmosis. It maintains osmotic balance, or you just remember that it maintains something to do with its own body. It maintains its own body's balance by letting water enter through its body by osmosis. Because animal cells have cell membrane, so to pass through cell membrane, definitely osmosis is taking place. The second point is about fishes and frogs. Fishes and frogs raise their osmotic pressure and hence absorb water from surroundings through osmosis. If osmotic pressure is increased, then more force will be applied through the water molecules, and hence water will be absorbed ultimately. And then the third important point also involves human beings. Land animals gain water by food and fluid intake and lose it through the body by the process of perspiration or respiration. Now, gaining of water and food is done by osmosis. Let me just write it down here so that you can understand. Gaining of water is done by osmosis. Done by osmosis. Okay, I just made a mistake here. I'm sorry. It's not gaining of water. Water is lost through perspiration or respiration. Perspiration or respiration involves osmosis here. So perspiration, because it is coming out of the skin cells, it is coming out of the skin cells by passing the semi-permeable membranes of the cells. Hence osmosis is taking place. That's why these two processes are going to require osmosis, involve osmosis for the removal of water. Those were all about the significance of osmosis in plants and animals. Now we are going to be discussing about our next topic of today's video, that is tonicity. So let us begin with the portion of tonicity. First of all, let us look into the definition of tonicity. What the definition says: the different concentrations of solutions which determines the direction of movement of solute or solvent particles during a process is called tonicity. Now what does that mean? You all can understand that solution means a mixture of solvent plus solute. So if this is a solution, suppose, let me just draw it out. This is a solution, and a solution always consists of both solute, sorry, sorry again. Okay, both solute plus solvent. Solute is the solid particles, solute is the solid particles, and solvent is the liquid medium, or you can say the liquid particles. Let us just stick with the liquid particles. All right. So together, the solid particles and the liquid particles make up a particular solution. That is what is called a solution. Now, a solution can have different concentrations. Like, suppose in solution 1, there can be more amount of solute and less amount of solvent, while in another solution, there can be more amount of solvent and less amount of solute, or vice versa. These actually together define the concentration of a particular solution. So the concentration of a solution means the amount of things that it has and the quantities in which the things are present. There can be 20 grams of solute and 50 ml of solvent, or there can be 50 grams of solute and 100 ml of solvent. These are certain configurations which define the concentrations of a particular solution. Now, what is tonicity? Tonicity is basically the difference in the solution, difference in the concentration of different solutions, which help us to determine the direction of the movement of solute or solvent particles. That is, for example, if inside a solvent a particular process is taking place, for example, diffusion or osmosis or anything, then based on the concentration of that particular solution, the things are going to move inside the solution, right? If you remember the process of diffusion, then it was a process in which substances were moving from the region of higher concentration to the region of lower concentration. This particular movement was possible because there was a difference in the concentration of both the mediums. That is what is tonicity. So tonicity tells us that how and in which way the solutions or the solute and solvent particles are going to move in a particular solution during any process. That is simply what is known as tonicity. Now, tonicity is of basically, okay, let me just erase that for you all. so that that portion is visible. Okay, tonicity is basically of three types. The first type is, let me write it for you all. The first type will be isotonic. The first type is isotonic. The second type is hypotonic. Sorry for that again. Okay, so the first type is isotonic. The second type is hypotonic. And the third type is hypertonic. Now, a very important thing that you all should remember is whenever in biology or in any science subject you get these terms which I'm encircling right now, then you need to know these meaning of the terms. Firstly, iso means same. Hypo means less. And hyper means more. If you remember the meaning of these important terms, then it will help you a lot in further studying of science. Iso means same. You all have heard this term iso in chemistry a lot of times. Isomerism, isomers, isobars, isotopes, all these things. That means they belong to same group. They belong to the same type. That's why the meaning of the term iso means same. Second is hypo. Anything hypo means less, and anything hyper means more. Now let us try to understand these three types of tonicity with the help of a particular diagram. We are going to be taking the help of this particular diagram. It will be easier for you all to understand this diagram. Okay, first let us understand isotonic solution. Isotonic, iso means same. Now, whenever you all will be studying tonicity, you all have to remember that here the surrounding of a cell matters a lot. So we will be considering the surrounding, the concentration of the cell surrounding in a very high amount. So let me just border the surrounding of the cell, and we are just going to consider this area for understanding what is isotonic or what does isotonic mean. By the diagram, I think you all can understand by now that this is a plant cell, a typical plant cell, because it has a vivid cell wall and a vivid cell membrane. So you all can understand well that this is a plant cell that we are talking about. Now what is being shown here? Here we have two arrows which is indicating the entry of H2O and as well as the exit of H2O. Both are involving H2O here. This is the vacuole of the plant. This is, okay, let me just change the ink of the color. Yeah, let me use red. Red will be easier for you to see. Okay, this is a vacuole. Vacuole contains sap and sap contains H2O. Obviously, H2 helps in the better mixing of all the solid minerals and everything. So H2 is a main ingredient, or you can say main part, constituent of the particular cell sap. Now, moving on with the concept of isotonic. Isotonic solution means that outside of the cell, that is the surrounding of the cell, and the inside of the cell, both the places are going to have the same concentration of solutions. So let us, for example, take and consider this cell to be a root cell. Roots are those parts of the plant which are present under the ground, and they are the one which perform absorption in plants. So we can pretty much say that this is the root cell that we're talking about. and in the root cell is surrounded by soil. Soil also consists of a concentration of solution in it. Hence we can say that roots absorb few substances from the soil. We have already learned about this. We have already discussed a lot of times about this thing. So isotonic solution means nothing, but it is a condition which defines that the concentration of the outside of a cell and the inside of a cell both are same. So in this kind of situation, no uh, no extra adding of substances are happening either out of the cell or inside the cell. So this particular thing is known as isotonic solution. Okay, I've removed all the ink on the slide for isotonic solution so that you can understand hypotonic solution well. So let us understand hypotonic solution now. If you remember the meaning of the term hypo, hypo means less. Less here, that means the surroundings. We are talking about the surroundings. I've already mentioned to you that while studying tonicity, you all have to keep in mind and take care of the surrounding concentration specially. So here there is uh, a discussion about the surrounding of the cell, and the cell contains less amount of solute. So the cell, that is the outside of the cell, contains less solute, more solvent. This is present in the outside of the cell. But inside the cell, just the opposite is present. Less solvent and more solute. Therefore, water is going to enter into the cell from the surroundings. This is water enters into the cell from the surrounding. This thing is known as hypotonic solution. So when the concentration of the surrounding of the cell consists of more solvent and less solute, then water enters from the surrounding into the cell. That is the concept of hypotonic solution. Now quickly understand what is hypertonic solution. It is just going to be the opposite of hypotonic solution. If you remember the meaning of the term hyper, it means more. That means the outside of the cell consists of more solute and less solvent, and the inside of the cell consists of more solvent and less solute. That is why what is going to happen is water will come out of the cell. You can see there are two arrows which are indicating exit of water. So exit water. I'm writing over the arrows. Water is going to go out of the cell, and the cell contents are going to shrink in this particular direction. From all sides, the cell contents are going to shrink. Okay. If you see these yellow lines, this yellow thing is basically the cytoplasm of the cell. Let me just mark it for you all. This is cytoplasm of the cell, and the cytoplasmic content of the cell is going to shrink inside, towards inside of the cell. This is what is known as hypertonic solution. Now let us quickly look into the definitions of the three solutions that we just now read. First, we have isotonic solution. When the concentration of water and solute is same, okay, this was supposed to be same, on either side of the cell membrane, that condition of the solution is called isotonic solution. Let me highlight the important words for you all in this particular definition. So when the concentration of water and solute is same on either side of the cell membrane, that condition is known as isotonic solution. So if the cell is in isotonic solution, then no change is going to take place inside the cell. That is, no entry or exit of water is going to take place. Going to happen. That is, we can say that no endosmosis or exosmosis taking place. Right? If you all remember the definition of endosmosis and exosmosis, which we discussed in the previous session, then endosmosis means when osmosis takes place and water goes inside the cell, that is endosmosis, and exosmosis means when water comes out of the cell through osmosis. So When the cell is in isotonic condition, no endosmosis, no endosmosis or exosmosis taking place. Let us move on to hypertonic solution. Hypertonic solution is basically when the solution outside the cell has higher amount of solute than the inside of the cell, then the condition of the cell is said to be hypertonic solution, right? That is simply what is known as hypertonic solution. We discussed about it. So let me just highlight the important words here. When the solution outside the cell has higher amount of solute than the inside of the cell, the condition is said to be hypertonic solution. That means in hypertonic solution, endosmosis and exosmosis is going to take place because this is a hypertonic solution. So only exosmosis is going to take place because water is going to come out of the cell. Is going to take place. If you remember the previous diagram in the previous slide, then in the diagram that was shown that the arrows were coming out of the cell. That means water was coming out of the cell. That means exosmosis was taking place. So exosmosis is going to take place here. That is water comes out. That is why exosmosis takes place. Just the opposite is going to happen in case of hypotonic solution. Let me just quickly highlight that for you all. When the solution outside the cell has lower solute than the solution inside the cell, then the concentration, then that particular condition is called hypotonic solution. Okay, here are few mistakes in the typing. I'll just correct them out. I'm sorry for the mistakes though. When the solution outside the cell has lower solute than the solution inside the cell. Hypo means less, hence less solute will be there. That is what is being said in the definition. That is all you need to know about all the three types of solutions. Okay, let me just quickly correct the mistakes that we have here. This is going to be that condition, and here endosmosis is going to take place. That is, water is going to enter. Endosmosis is going to take place and water goes inside. Acha, one more very small thing I need to mention to you all. In case of hypertonic solution, you all have noticed the previous diagram, the cell contents were shrinking. So that means that hypertonic condition is going to involve the shrinkage of cell. I'm going to write it for you all. Though it is a very easy thing you all can remember yourself, but still I'll write it so that you never forget. Hypertonic solution is going to involves shell shrinkage. That is, the cell contents were shrinking towards the inside. And hypotonic solution is going to involves swelling of cell. That is, the cell is going to be puffy after absorbing a lot of water inside it. That is all about tonicity. That is all about the three types of solutions, and that is all you need to know about this topic. So if you have understood the topic, then make sure you just like the video and share it with your friends as well. We have already finished all the portions that we needed to discuss in today's video. So we have come to an end of today's portion. Please definitely share this video with your friends if you have found out to be useful. And if you have any doubt in any question regarding this particular portion of today's or regarding the whole chapter, then please do not hesitate to write it down in the comment section. I'll definitely answer them. Also, make sure you subscribe to my channel and stay tuned for the next videos, upcoming videos. Till then, Take care of yourself. Stay safe. Stay happy. Peace.